Just thought as we prepare for the next program part here, which we called Flash Talks. So just that you get an idea why we have that here. We had that similarly in the first conference too. We think it's very important to have young researchers, namely PhD, postdoc level, as part of this conference. Very, very important that they share their research and their questions and their projects with us. So this is why we invited for altogether four Flash Talks, young researchers, from many different countries, and very excited to welcome the first, more or less, panel. And we have a moderator of this, and this is Maurizio Muraca. Maurizio, may I please hand over to you?
So our first speaker is Dr. Gaetano Granatelli from the University of Augsburg. And the title of his talk is The Theory of RNA Replication and Evolution in Phase-Separated Systems.
Thanks for the doctor, but I'm not yet a doctor. Morning, everybody. My name is Gaetano, and I am a PhD student in theoretical biophysics at the University of Augsburg. And I'm here with my friend and colleague Samuel. So I would like to start my talk with a simple question. Why a theory of RNA replication and evolution in phase-separated systems is an exciting topic to discuss? And the reason lies in these following slides. So synthetic life is a grand challenge driven by the pursuit of unraveling its fundamental principles and the prospect of developing innovative life forms harnessed for practical purposes, like this one. This is supposed to be a synthetic cell. It's not a potato. And one way or another, the starting point for such a challenge is life as we know it. Nowadays, we know that all non-life forms trace back to a last universal common ancestor, better known as a LUCA, that witnessed the onset of Darwinian evolution and eventually led to the diversity and the richness that nowadays we can see in the tree of life. But how did we get to LUCA? The Origins of Life community believes that at the culmination of a so-called molecular Darwinian evolution process, a molecule or a set of molecules with self-replicating abilities has evolved to become LUCA. And now an interesting question we could start asking ourselves is, what do we need for a synthetic LUCA? That means, what kind of synthetic self-replicating molecular system could showcase an evolutionary ability such to give us a new LUCA again? And now with this bigger picture in our mind, my talk can start. And please emphasis on the adjective bigger, since of course I'm not going to give any answer to such a big question. I'm just going to propose an exciting model from a very specific angle. Now, before getting to the details of the model, a few more comments related to this bigger picture I just introduced. First of all, it's important to highlight that such an ambitious goal constitutes just one side of the coin. The other being this dual aspect of ethical issues and responsibilities. And I'm aware that we're going to discuss this extensively during this talk, as we already did. So I will allow myself only to share with you all a short thought about responsibilities from a theoretical physicist perspective. And we believe that as one scientific community, we should all share the ethical responsibilities across the disciplines, instead of, let's say, putting them just on whoever one day will create the experimental conditions for synthetic life to be engineered. Next, what's the state of the art about synthetic life? So it's quite obvious to say that such a grand challenge requires unified approaches that go beyond individual disciplines. Usually, two main approaches are used across different fields, the top-down and the bottom-up approach. For example, the model I'm going to present conceptually stems from a bottom-up approach, which is a strategy where the process begins at the most detailed and basic level and works upwards to progressively build the complexity and a more comprehensive picture. A different strategy would be the top-down approach, of which my friend and colleague Samuel will talk later for a different case study. And currently, a lot of fields have shown considerable progress toward the engineering of lifelike systems. And although it's quite obvious to say that in practice, the experimental disciplines will be the ones which one day will provide these conditions for the engineering of synthetic life. We strongly believe that the role of theoretical biophysics, and more in general, any theoretical discipline, is essential for such a collective effort. And this is mainly due to two key strengths of the theoretical disciplines. On one hand, the ability to develop theoretical and computational descriptions of extremely complex phenomena which often cannot be studied experimentally. And this can help in how to interpret increasingly complex parameter spaces, but also in how to guide maybe the design of future experiments. And second of all, the ability to extract general principles and generate testable predictions. And this, we think, it's an essential predictive power for triggering upstream ethical discussions. And now, with all this being said, let's finally move to the model. And let me start with a very short crash course of a couple of concepts we need to understand the model. The ability to evolve, the so-called Darwinian evolution, is agreed to be a key feature which distinguishes living things from non-living molecules. And it has produced a diverse and complex living world. We know that evolution is due to an error-prone replication process of the genetic material, the genotype. And such replication process is performed by some replication machinery, which, according to the central dogma, is transcribed and translated from the same genetic material. The replication intrinsically yields mutations, leading to a set of genotypic variants which are propagated to the copies and eventually affect the so-called system observable traits, the so-called phenotype, which in this case is represented as different variations of the same protein. And then we know that the interaction with an environment endows each phenotype with a given fitness, which broadly is defined as an organism's ability to compete, survive, and reproduce. And eventually, the mechanism of natural selection makes sure that the copies with a fitter phenotype will thrive at the expense of others, since all of them compete for the same resources. And now from this picture, it's quite clear that a living entity must be able to replicate. So it must autonomously make a copy of its information storing substrate, and also, of course, also to ensure that all other critical components must be also self-synthesized. However, what is still unsure and unclear is how this process works at the most primitive level. That means how molecular Darwinian evolution is able to develop in the first place the complexity of some primitive self-replicating molecular system. For example, our model assumes the self-replication ability. So we assume that molecular evolution already developed molecules which can promote their own replication. And what we're actually assuming with this in detail is that evolution already provided some form of selection mechanism guiding some single nucleotides to form sequences which are functional, meaning that the sequences can assume a 3D conformation which is catalytically active. And then what our model tries to investigate is therefore not the emergence of the self-replication ability, that would be a different model, but rather how interactions among self-replicating molecules could build up the complexity and lead to some form of coevolution. And here we are with the main components of our model. So we have a substrate, which is a set of monomeric subunits. And then the model takes into account a full sequence space of equally length precursors, which we define as RNA-like molecules able to self-replicate with moderate accuracy. Each precursor sequence can encode the information for a replicase, which is the catalyst of this guy's replication. And in particular, we assume that only those precursors containing a more or less preserved motif, which is just a pattern of monomers, these ones can keep the replicase formation ability. So for example, if we have a precursor that, because of mutations in the replication, alters too much this motif, and this can lead eventually to lose the replicase formation ability. And these precursors are called parasites. We have different mechanisms of replicase formation, and finally we also introduce a sequence-dependent feedback in the replication through the binding of complex and precursor of replicases and precursor, forming the complexes. And now, with such a model, I would just like to conclude my talk with what kind of questions can we answer with a model as such. And for example, given a sequence space that we define, given a replicase encoding motif, we could initialize our system with a given distribution of precursors, replicases, and complexes. And of course, we also need to introduce some form of metric to quantify the number of mutations with respect to this motif, but as well some key observables to quantify the fidelity of the replication process of our precursors. And with the model as such, for example, we could start asking about the coevolution of precursors. So for example, if there are any parameters that drive to extinction the precursors, or for example, that can ensure a steady growth of them. And this is our specific angle in the group, to wonder about the influence of especially heterogeneous environments. And with this, I would like to conclude my talk, and I thank you all for the attention, and I'm happy to take any questions. Thank you.
Thank you. Congratulations for addressing such a complex issue. There is time for just a very short question.
Just a quick question. Quick question on the assumptions. When you look at the materials that are needed for the precursor and your replicase, right? Mm-hmm. So what kind of parameters do you have to have in place for that to make all the rest of this successful?
So, for example, the model is quite agnostic to such a choice. For example, we know that life and biology works with this 4 letters alphabet. In principle, for example, what I'm practically doing is reduce the complexity and use a binary alphabet, so 0 and 1. This does not affect the whatever conclusion we can take from the model. Yeah.
Thank you.
You're welcome.
Thank you.
Thank you.
Our next speaker is Dr. Eric Poppleton from the Max Planck Institute for Medical Research in Germany. The title of his presentation is Building Biomimetic Hardware for Synthetic Cells.
Fantastic. All right. Hello, everybody. My name is Eric. I'm a postdoctoral fellow at the Max Planck Institute for Medical Research and many other institutes around Heidelberg. So basically what I want to talk about today kind of really follows up on what we just talked about, which is how do we actually build the components that we could do bottom-up synthetic biology with? So if we think about cells, they're really complicated, even at the most simple level. Some of you may have heard of this minimal cell project where a group at Harvard has been trying to strip down a cell into the very simplest components and try to minimize its genome as much as they could. And what they found is that they still needed somewhere between 100 and 200 genes, some of which they didn't know what they were, in order to have cellular function. And so if we look at this artist rendition of the minimal cell, what we still have a transcription and translation system, which is over 150 genes, stored in a spaghetti mess of DNA and proteins attaching to that, and RNA coming off the DNA, then translating into proteins. And if you look at the cytosol, it's crowded with proteins just engaging in metabolism, engaging in signaling. There's a lot of stuff happening. Stuff has to diffuse through the cytosol. Things have to find their binding partners. It's all a very complex, noisy system. And to top it all off, we also have the membrane. And membranes are what are protecting the cell from the outside environment, but they also give you a platform for which proteins are able to interact with and engage with the outside environment, either introducing signals, taking up metabolites, or expelling waste. So anyway, this is a very complicated system that has many layers to it. And everything that we do here has been affected by 4 billion years of evolution, which kind of puts noise in the signal. This is not a simple system where you can look at something and go like, that does that. Instead, what you end up with is something where everything is interacting with everything else. Everything kind of matters in some way, and trying to find the signal from the noise can get very difficult. So if we're interested in the basic principles of life, why don't we ask ourselves, why don't we just start again? What could we learn from a lifelike system if we were able to start from just a simple lipid bilayer and start trying to put things inside of the bilayer? So for example, what if we start putting in membrane pores? So people have done this with DNA nanotech before, showing that if you introduce a DNA pore, you can introduce either size or chemical specificity into a bilayer. What can we learn from that? What can we learn from being able to put in structural proteins? You know, cells have these very fascinating shapes that allow them to interact with their environments, to move. But this all comes from the dynamics of various structural proteins that are pushing and pulling on the membrane in various ways. And the group that I'm a part of, Kirsten Gutfrich's lab, has done a lot of work on using DNA and RNA nanotech to build filaments that are able to deform membranes and move towards things like cell division and cell morphology from a very minimal perspective. Finally, what if you did things like Petroswela's lab does, where they have they have protein-protein interactions, which are then on a membrane, and you can get these waves of propagating signal across the membrane surface. And this is all based on the way that E. coli cells find where their midpoint is when they want to divide. But we can also recapitulate these signaling cascades in these simplest possible ways in order to understand the basic fundamental properties that make signaling possible in biological environments. So remember what I said before. It took over 150 genes to build up a minimal transcription and translation system, which means that producing protein components is something that requires a ton of overhead. There's a ton of basic components that you have to have in your system before you can have something that looks like life as we know it today. And as we saw in the last talk, everything that we know comes from Luca. It all uses this same underlying machinery of ribosomes and the whole central dogma. And so what if we could do something that was a little bit simpler, that didn't have this overhead? So what we're trying to do right now is, what if we could build life just from RNA? I mean, you've maybe heard of the RNA world hypothesis, this idea that maybe life started with just RNA. So we're kind of trying to take us back to that. Can we build up a much simpler system that only uses RNA, that is able to build out lifelike function without having to rely on this enormously complicated transcription translation machinery? Some advantages of this. Number one is we still have one protein we have to get in there that we've not found a way to get around. And this is some sort of polymerase that is able to take a DNA template and make RNA out of it. So we're down to one protein from 150, not bad. But the nice thing about RNA is that it can also do a lot of the things that proteins does, just worse. So aptamers are able to bind targets. They're able to change the structure of the RNA and transduce signals. And ribozymes are able to do somewhat complicated enzymatic functions. In this case, what I'm showing here is a self-splicing RNA that is able to cut a substrate sequence. And therefore, you're able to do something that maybe approaches division. So what exactly am I working on? I'm working on how can we build membrane pores specifically. So the idea of RNA origami is that we can do rational design of DNA and RNA structures, and then we can use that to build up nanoscale objects that we are able to position chemical groups on. This all comes from a 1982 paper where Ned Seaman pointed out that, hey, if you take a DNA junction and you start connecting these together, you can use this as a lattice, and by controlling the angle of those junctions, you can build up essentially any arbitrary 3D structure. What you can do is you can use that to build up a schematic of the thing that we're going to make. So this is the schematic of my pore. And then we can express that. We can do cryo-EM on it and see that, yes, in fact, we do get a pore-like structure with binding overhangs here that we can use to functionalize with ways to interact with membranes. Now, the thing that I'm skipping over here is how exactly we do that. But suffice it to say, we have a membrane-binding RNA aptamer, so we're able to induce affinity for the membrane. And then we can do single-channel ionic current recordings where we measure pores forming in very small membranes and be able to see that, yes, in fact, we do see current jumps corresponding to the size of the pore that we expect to see. This is still an ongoing engineering project, and trying to understand why this thing even works is also a very ongoing project. That is what I spend most of my time on. But I just wanted to give you guys a flavor of some of the things that we are trying to build from the bottom up in order to make a minimal cell. So kind of since we're at an ethics conference, I wanted to throw out this idea of responsibility, because I believe that is the topic of the next panel discussion. So what is our responsibility as molecular engineers? One of the advantages of bottom-up synthetic biology as opposed to top-down is that it is a more interpretable process. But despite this, unexpected behaviors may still emerge. And so it kind of it's important for us to ask ourselves, like, what is our tolerance for unexpected properties, and how much do we have to understand a system before we are willing to push it forward and use it for functional purposes? Second, top-down systems, we might say they're like more dangerous to biology. If we're using like a CRISPR system, for example, we always talk about this danger of off-target cuts and off-target mutations because it is able to interact directly with biology. But they're also more susceptible to biological control systems. So for example, if you're making a synthetic microbe, you can still, and your synthetic microbe gets out of control, you can still kill it with any biocide. We don't necessarily have that for bottom-up systems. In this case, we're working with RNA, so you still have some controls, but if you were to make something that is totally synthetic, you lose those controls that we have built up and that biology provides us for controlling systems. And finally, who has the responsibility for making these ethical decisions? Here we're a group of scientists and ethicists, and we kind of have this tension in the room between scientists who may have a deeper understanding, but we also have this kind of selfish incentive to push our field forward, both for our own curiosity and also for our own funding opportunities. So just some things to think about in the next panel discussion. And with that, I won't hide you guys from lunch. I'd like to thank the two groups I'm a part of. So BPE does all the laboratory work, and MBM does a lot of molecular simulation. So as we try to build up models that can understand why these things work. I'd like to thank my funding agencies, and that is it. Thank you. Questions?
Thank you for your presentations and your questions. A little bit frightening. Any quick comment? One quick comment or question?
So thank you for the talk, and especially at the end, pointing out sort of the assumption that you have based in this thing is that you're building it on what we already know, right? Mm-hmm. So is there any impetus to look at, say, a silica-based system or something totally different than what we know currently as life?
So you're kind of get to this challenge of we can build much more complex things if we take life as we know it. And if you start again, you really have to start again. And so you get stuck in this like, okay, we can start building something that vaguely looks lifelike out of silica, but you have to do all the engineering from scratch and you don't get very far. So our approach has kind of been this happy medium where like we're taking the molecules out of biology and then making them do things they were never meant to do. Like DNA and RNA origami is really hard to explain to people sometimes because it's not information anymore. We are using nucleic acids as structure. But at the same time, we're still taking life's molecules because we know how to work with them. We have tools for them. If I want a big DNA sequence, I can go over to the molecular biologist and get something cloned. I'm not so stuck with trying to reinvent the wheel.
Thank you.
Thank you. Thank you to all the speakers and participants. Small panel, but very big questions. Thank you.
Housekeeping.